Welcome, everyone. Before we begin, I would like to say a huge thank you to Litmaps, which is our first official sponsor of the podcast. Litmaps is a wonderful company I've been blessed to collaborate with for the last couple of months. And what Litmaps essentially allows you to do is to choose your selected papers, whatever research field you are in, paste it into Litmaps, and get a beautiful, wonderful map, a visualization of how those papers are connecting to the rest of the field. So one particular use case I found to be super useful at the beginning of my PhD when I was just entering this field of neuroimmunology was to get a broad overview of the field. So what I did was I found the biggest review I could find that was written in 2007 by the leaders of the field. I pasted it into Litmaps, and it gave me this wonderful map of all of the papers that are heavily citing my review, but that have come out since the last 15 years. So I got my beautiful big review plus all of the updates. Then I click monitor, and now I get updates on all of the papers that come out but are super relevant to what I'm looking for. So check out Litmaps. They're awesome. Their new user interface is also very nice. Super easy to use. There's a free option. If you want to upgrade to the paid option, you can do that. But by all means, you're not obligated to do so, and you can still get tons for free from Litmaps. So the link is in the description. Check them out, and let's get on with the podcast. Well, all right, everybody. I've been chatting for the last 15 minutes with Elena Hoffer. She is a Ph.D. in Immunology from Karolinska Institute, and recently she started her own company called Alma-Me, which helps PhDs transition to industry. And I really hope we can have an interesting conversation. This is a topic that I'm not so familiar with. I know people talk about it a lot, transitioning to industry. It's many people's dream as a PhD. But today I'd like to pick your mind on that and many other topics. But for the beginning, maybe Elena, first of all, welcome. Thank you for agreeing to talk to me. Maybe tell us a little bit about your path to KI, your background, your education, and so forth.
Perfect. Thank you so much, Kenan, for a very nice introduction, and thanks for the invite. I'm super happy to be here and speak to you finally after we have talked on LinkedIn for quite some time. So who am I and how did I make it to KI? I think I had a quite non-linear path. So I want to share this because I think this is a thing for a lot of people, maybe that they're worried about I'm too old or God knows what. Can I do a PhD? Yes, you can. I'm 34 now. I finished last year, so it's never too late. But so my path went a bit different. Like after high school, I did an apprenticeship as a pharmaceutical technical assistant. So I worked in a pharmacy for half a year. After that, I moved to industry and I worked in a lab. And, it was this type of job where you had a protocol and you followed the protocol. And I was like, oh my God, this is boring. I need to get out of here. So I was like, okay, I'm going to go and I'm going to study. So I was always interested in, pharmaceuticals and how it just amazed me how, you take a drug and it can kind of change things within your body and how things work and how you perceive things. And so this is why I went for the pharmaceutical technical assistant, because it's a lot about how work drugs and what do they do in the body and when do you take what. And so I went on to study pharmaceutical biotechnology in my undergrad in Biberach in Germany, which I really loved. It was quite technical, more on the engineering side. It was a lot about, how you actually, how do you produce antibodies as a drug? And I really loved it. It was so much fun, like all the different things that we went through from manipulating, cells and giving them information and kind of manipulate them that they produce something and how you culture them and how you actually at the end, you get the whole of how you actually clean it up. So it was just amazing to me because it was something that you cannot see with your naked eye, but it's somehow happening. And I was just always amazed by it. And then I actually did my bachelor thesis 2015 already at Karolinska Institute. I was really interested in vaccines. And so I went there and I worked in a lab where I could work with vaccines and vaccine adjuvants.
How did you sorry to cut in, how did you get to KI at that point? Because you were in Germany and then you go to KI for your bachelor thesis. How was that process for you?
So to be honest, I was like, okay, I want to go abroad because I kind of wanted to challenge myself a little bit. It was very far out of my comfort zone. But one of our professors, he did a PhD at Karolinska Institute. And Karolinska Institute was an option for me because it's in Sweden. Everybody speaks English, like versus if you go, to Italy or Spain or all these other European countries, it would maybe be difficult to survive only with English. So because my professor did a Ph.D. at Karolinska, he was advertising it. So I just actually started looking up groups, and then I saw that some groups, they actually write that they're open for getting students. And what I did, I just wrote an email, honestly, and I think it was actually not even a great email. But yeah, I mean, actually, I got the job, and it was so funny because this woman that took me in 2015, she became my co-supervisor, and at my defense in November 23 now, she actually printed the email and read it for me, how I ended up with her. And it was like, I was ashamed. Not ashamed, but I've never, I'm not a language person, so my English was very different at the time. And like, it's just, it was just a very funny email looking back. And I would never write it like this again, but there was something that actually caught her eye. And yeah, I got the position. So I think honestly, if you're a human being and you just reach out and you somehow manage to catch people's eyes or show them what you can bring, I think it's actually not that difficult to receive a response. But I also think a professor needs to be in the state where they want to take students in. So if they don't need anyone, they're probably not going to read it, or maybe they read it and they just delete it. I mean, based on my experience. But I would just go for it and try it. You have nothing to lose at the end.
Yeah, exactly. I think 2 points there. And every time I say 2 points there, I start with the first point and then I forget the second one. But 2 points there, and I'll try to remember both of them. One thing is that students that I work with often that are looking for positions for a master's or a PhD or whatever, often take these applications as something non-personal, as if you're being dealt, with a computer. But in the end, behind those applications, is a human, right? So sometimes things have to match, of course, and that's the point number 2. So I don't forget. So they do have to be in a position that they're receiving students, and that's why you shouldn't take it personal if they don't, right? Because sometimes just things don't match in academia many times. But I think that's a very crucial point you mentioned there. Sometimes if you just ask, you will get what you ask, because you're asking another human being, and if there's a common interest, and if there's a connection there, and if you can bring something to the lab and you can show it, sometimes life will give you good lemons. All right, so you get to KI. So tell me also what was wrong with that email besides English.
No, nothing was wrong with that email, but because coming from Germany, and then you write this email. Also, there was no ChatGPT, guys, because ChatGPT would have probably helped me. This was in 2015, and I wrote this email, and I think I didn't ask anyone for help to help me, but actually I think I said your research interest aroused my interest, or your research topic aroused, and aroused is just the wrong word. But the funny thing is, if you translate it back into German, it would actually work. It was just it was just funny. I think that was the most funny thing in the email. The way it was just the words that I used, but it was. At the end, I mean, she chose me. I managed to get her interest in.
And she was probably reading, being like, "Oh, this person's really excited. She's aroused."
I think honestly, this is actually what Kinan. I think this is actually what she said, because she read it at my defense. Because in Sweden, when you defend, you have this big dinner at the end of the day, and it's like a big party with I had 60 or 70 people, and then everyone gives a speech. And since she was my co-supervisor, she read it, and I think she felt like I was like. I mean, it was funny because when she read it, she was just laughing, because I think this is exactly what she said. She was like, "Oh my God, this person is really excited working with me. Let's see what she can bring to the table." And we had a chat, and I think you had a really important point, Kinan, and I think this is something people need to remember. Just reach out. We're all human beings, right? I mean, I do see that you have this hierarchical type of thinking, but at the end of the day, I mean, if you approach people like human beings, most of the time, honestly, I think you will receive a response, because it's just the way it goes. And I guess you have nothing to lose, and I think you should just not get discouraged when people don't reply. It really rarely has something to do with you, rather than maybe the timing or something. They cannot always take someone in because there is no one who can teach you, because this is something you should also remember taking someone in because of your responsibility to teach. It's not only that you bring actually something to the lab and you help them fixing something. They also need to have they need to invest in a huge amount of time and resources.
I do want to just share one quick anecdote before we move on from that. So there's just so many reasons why someone wouldn't respond to your email, and sometimes it can be personal, like I just don't want this person to work with. But most of the time, it's just people didn't see the email because they're overwhelmed, or they read it but they just have too many candidates, they don't know what to do with it, stuff like that. So one anecdote from my side that might encourage people to reach out to professors more or to companies more is when I was applying to my master thesis at Harvard, I initially had an interview and it went great, amazing, you can come to the lab, great. And then after that, I was super excited and I sent a follow-up email. Like, thank you. Can we fix some details, some dates? Like when would I exactly come? How would we do it? Can we think about the topic? And I get no response. So I wait about a week, still no response. Okay, so I follow up again. Hey, just in case you didn't see my email last week, I repeat this process a couple of times. No response. Now it's been a month and a half and I'm ready to give up. Like, okay, this person maybe doesn't really want me, or they lost interest, or just I should just move on maybe. But then I think, at the time I was at the University of Heidelberg in Germany, and we were using this very old email system. And I remember some people telling me that it was bugging and sometimes it wasn't delivering messages. So I tried, okay, maybe if I just send from a different email, just one last shot. So I sent it from my other university, from my bachelor's university email. And the professor replied within 30 minutes, said, "Oh, I was wondering where you disappeared. Why are you not coming to the lab?" So I almost missed my opportunity to go to Harvard because, I thought, there's something personal there when actually it was just a bug in the system. And that happens maybe more often than people think. So, all right. So you get to KI, you do your thesis. What's happening next in your journey?
I went back to Germany and I actually did a master's in pharmaceutical biotechnology. So I just kept going because it was like something I was really interested in. And this time it was at the University of Ulm in collaboration with University of Biberach, so still in Germany. And then when I approached my master thesis, I was like, I really like Sweden. I really like Stockholm. I fell in love with Stockholm. It's a beautiful city. So let's go back. And I had to tell you like this, I don't know. And I mean, you moved a lot too in your life, but for me, it's like, building a life in a new city, it always takes a lot of energy. Not just like, like you mentioned earlier in our chat, finding an apartment, but also like finding a new circle of friends, setting everything up for your life, getting familiar with the whole system. So I was like, okay, you know what? I don't want to go into a different country. I really like Stockholm and I kind of really know how things work here and I know people that can help me. So I applied at Karolinska, I actually reached out to Karin at the time, who I did the bachelor thesis with, and she didn't need anyone at the time. But then she said, I know a person who would actually be interested potentially. So she referred me to that new person who then later became my PhD supervisor. So I did my master thesis then in the lab where I also was over the PhD position. So this is how I came to move from a more engineering background, which I had with pharmaceutical biotechnology, into like really hardcore T-cell biology and immunology. So I completely switched gears, and I felt at times really, really lost, I have to tell you, Kinan. It was a tough transition when you go in and you're like, okay, the only lecture I ever had about immunology in my undergrad or during my master's was about, it was pharmaceutical immunology. So it was really more about how can you modify antibodies so they can become small and actually penetrate the brain. So this is the only immunology. I mean, of course, I knew, from high school, there's the innate and an adaptive immune system, but I was basically not an expert at all in whatever I kind of signed up to do a PhD in.
So tell me about that beginning, because I'm now in the beginning of my PhD, and last week I also had a conversation where I related to, beginning suffering of a PhD student. So tell me about that beginning. Okay, you had a little bit of background on immunology, but it's not enough, because now you're expected in the next couple of years to produce new knowledge, to get to the edge of knowledge in that little field, wherever you entered, and produce something new for other experts to see and value. So, but you're beginning from just having one or two courses on it. So tell me, how did you feel in the beginning? What were some things you were facing with?
To be honest, in the beginning, I felt pretty like a real, I don't want to say dumb, but I mean, I didn't know much, right? And I just saw all these people around me. And then I was this person, I compared myself to PhD students in the lab that were like in their third year. And I was like, they know all of this. But I feel like, because I always felt like, I was a bit behind with my knowledge, I worked extra hard to acquire new knowledge relatively fast. So the way I did it, I mean, honestly, I read up a lot. I talked a lot to colleagues. So for me, it's really, I also loved, sitting in lab meetings, really listening and learning. I think that was very effective for me. But overall, I mean, you cannot really get around the reading part and just, kind of understanding. But also what you will see when I read, it's hard to make all the connections in the field. Now, 5 years later, I read the same papers and I read them with a very different, type of set of classes, and I extract very different information. But I would say it was really, really hard for me, Kinan, because I had no idea about T cells, all the different CDs, CD3, CD4, whatever. This was all so new to me. And yeah, but the thing is, like, I think I was also kind in a way to myself and said, okay, it's okay. There is a learning curve to everything you do. When you start a PhD, it's overwhelming. I was so overwhelmed. I cannot even tell you. Like, I had like so many moments, because from, setting up experiments to not really understanding the subject matter that you're working in, and like having all these connections. But I also have to say, since I did my master thesis in this group already, I had a very light start for my PhD, because I did have a PhD student that worked with me during my master thesis, and she was incredible. She took so much time to teach me, and she took so much time to explain things to me. And like, this was an incredible help, which really gave me a soft start in my PhD, because when I started my PhD, Kenan, I knew all the methods in the lab. So, from flow cytometry to imaging, like immunofluorescence or other sort of assays that we used. So the only thing I really set up was single cell sequencing, which was a big deal for me too, to be honest, to get all of this stuff right. But so I think starting the PhD was a bit easier. And I think at the time of when I started my PhD, I had a relatively good understanding of the field. I knew quite a lot about T cells and skin disease, because this is really like what I work in, because I had these 9 months where I could learn, and people really were, they supported me in my learning process. And also what helped me a lot, that I was presenting a lot in lab meetings, and I'm an overachiever, so and I do believe I'm a good presenter. So sometimes I just, I remember this one presentation where I talked about how does PMA iomycin, I don't know if you know it, it's a stimulant for T cells that we use for ex vivo assays. And I just, talked about how it works, and it was amazing, because I felt like I could teach them something. Because the funny thing is, sometimes you're in the field and you never question things anymore, because when you're so far ahead, you just do it and you just take it. Oh, this is how we have done it all the time. And no one actually wonders, how does it work? What does it actually do? Why do we use these substances to do certain things? And I think I was just always curious to answer these questions. And I was given a stage where I could present it, and I was always looked at as an equal. And I think that helped me a lot. I was never looked at as a master student. I was always, people always really, they gave me the feeling of being equal and not any less, because I'm not a PhD or not a postdoc. And I think it helped me a lot in, this believing in myself. And my PI, I had such a good relationship with her, and it was just, I felt very, very supported by her.
Was there a
Sorry, that was a
moment?
very long answer.
No. Keep talking. That's great. Was there a point in your PhD that you felt kind of a click, that the confidence is now coming a little bit and things are starting to come together, and maybe I know what I'm doing a little bit now, and I'm a bit of an expert, and I can do this?
Most certainly, but I never had that click because I think for me it was more of a gradual process. You learn, and I think at some point you just realize, oh wow, I actually know so many things. But you know what is also the thing with the gradual process? Because it goes so slow, you just take it for granted.
Like
It's not like,
aging, right?
yeah, exactly.
Yeah, every
It's
day you're the same, but
yeah.
then you look back and man, I have aged.
Yeah,
This is
yeah, exactly. So it's like, and it's so I don't think I had this moment, but I do think looking back, and I think this is important when you do a PhD, never compare yourself with other people. Always compare yourself to your own yesterday or a week before, or me, myself, and I like a year ago, and see actually what is the progress I have made as a person, and not like other people around. And I think I struggled a lot with this. I am comparing myself all the time. I always compared myself with people that were 3 years in their PhD, but also it motivated me to be better. So I don't think it's necessarily always a bad thing, because it gives you this inner drive to put in the extra step to get somewhere. Yeah. So
there's one thing
I think
I want to probe just a little bit. You mentioned one very interesting thing that I also find, which is that some of this knowledge about techniques, essays, whatever you're doing in the lab, it just accrues so much that people stop questioning it. And then it sort of gets relegated to, okay, this is just how we do it. How much of that do you think is, first of all, necessary to question to do good science? Or should you always just if you start questioning everything, then you kind of end up in a rabbit hole, and you have to go down to the first principles of every single thing, and that can drive you insane sometimes because you just have to finish this experiment. So how much of that is necessary to do good science? Or if I reverse that question, how much of not questioning that is maybe producing bad results in science?
okay, that's a very big question, and I actually need to think. So I think in general, Keenan, I always liked to understand the things I did. So for me, it's just like, if I don't understand what's happening, I cannot work with it. This is just the way my brain works. For me, I rarely want to take things just, this is how it is, because I know there is some sort of explanation. Why are we doing it like this? So it's just like, for example, you can run a single cell sequencing protocol. You don't need to understand what is actually happening. You just follow the steps, or you actually can start to try to understand. But with this understanding, I think it's important for me, and it's not necessarily about questioning, it's about having an understanding. So what happens if something goes wrong at this point, and how it actually affects down the line the experiment. So for example, coming back to the PMI iomycin example, if you know what each of these substances does actually and how it activates the cell, you will understand what happens if I forget to add iomycin, right? You will know the cell will not be activated because they only work together. So I think it's about, you don't need to question, in my opinion, everything, but I do think what is important is whatever you work with, have a basic understanding of how it works, because it will help you evaluate when you reach a point where something went wrong, where you maybe forgot something, where you used a different concentration. You can make some sort of interference of how it actually affected your experiment. So I think that's just important. I do agree with you. If you question everything, where would we get? I sometimes do also believe in academia, we keep doing things the way we have always done it. We have set up this protocol 10 years ago. This is how we do it. And this is just how we keep doing it. And if things work, it's great. But sometimes I also think, just because someone thought 10 years ago, this is the way to do it, technology is also moving quite quick. So I think it's kind of good to sometimes just see, is there a different way of doing it today versus, 10 years ago? Is there a more efficient way to do it? But I do think you need to find a balance, in my opinion, between, you cannot go in and reinvent everything and, try to optimize every little thing that is already established, because I think you have to find, a balance between, is it really worth it? Will the result, if I, for example, optimize this method with I'm trying a different thing and I want to see if I get more cells, will it at the end make a difference? Does it make a difference for me if I have 10,000 cells or 15,000 or 20,000? If the answer is no, then just go with whatever you have. I think it's all a bit, it's all about the context too. So it's not
So
a
it's
very,
kind of an investment, right? To a certain degree, and it's an investment with diminishing returns, right? To one point, you want to invest in understanding the method. So later, if something goes wrong, you can troubleshoot, solve it. But at some point, it's just not giving you a return on it, right? So you cannot reinvent everything, as you said. I did notice, however, that if you are new in a lab, and I've been a new person in quite a few labs, if you keep asking, why do you do it that way? People don't like you.
yeah, because they most of the time don't have the answers. Believe me, that's the thing, and I don't know.
That's how we do
because
it,
we
and
have
just
always
shut
done
up.
it. Yeah. Don't ask. Yeah, exactly. For me, this is always like, I'd never understood it, and I felt like, I can tell you a little anecdote. I never understood. I was like, when I joined as a master's student, I was so curious to understand everything, and I was like, how can you not know this? Working with this day by day. But what I also realized, when you do your PhD, you have different levels of priorities. So in the beginning, maybe you have more time to read, and as you move further in, you have other pressures. So at some point, you might just do things, and you might not question them as much anymore, because simply thinking about all the other things, do you have really time to understand it at this given moment, or do I actually need to do something else? So it's all about, people are not ignorant. I think it just breaks down, what can I actually do with the time that is given to me? And then you need to make a decision, what will make a significant difference? So I think it's quite, I don't know if this makes any sense. It's like, because when I was a master student, I had all the time in the world. I could read up on everything, it was just a very different commitment. And then when I went into the PhD and you realized, okay, now you have to manage everything yourself. It's just, there was simply not enough time to understand everything in detail as I had in my master's. So this is the time where I realized at the end of my PhD, and I reflected back and was like, when I asked people at the stage where they were, sometimes I asked them questions and they didn't really have an answer, and was like, that's interesting to me, because I feel like, I would ask myself, why is it like this? But I found myself towards the end of my PhD that I would have been in the same situation. You would have come to me and you would have asked me something, and I could not have explained it at this point anymore, because there was simply not enough time to dig into everything very, very deep. And then you start prioritizing things over other things. And most of the time, it's unfortunately getting experiments done rather than always understanding things in very depth. I don't know. This is my experience. Because, your cells, you have to take care of them. When patient samples come in, this is what your priority is. It doesn't really matter if you think, I should be reading about this. If I have a patient sample, I need to take the patient sample. And then you have only X numbers of hours in a day. So, yeah.
How would you describe, what did you study during your PhD? Okay, so it's immunology. It's something to do with T cells. But if you can zoom out on your whole PhD journey, what did you study, and maybe share, what did you discover during that time?
Okay, so I will talk about my main project, because this really is my baby. It's not published yet, so I have worked my entire PhD on it, and so I will start a bit earlier. So what was the project for my PhD? So we work in the group where I was. My boss, she's a dermatologist and scientist, and she's interested in skin diseases like psoriasis, for example, or also vitiligo. What is interesting with psoriasis, it's a T cell driven disease. So imagine you have this little psoriasis flare here, and then when you treat patients, it would disappear. But the interesting thing is, when you withdraw the treatment, the psoriasis will come back at the exact same spot, indicating that there is some sort of memory. So we think, or it's quite known, that T cells play a pivotal role in the reinitiation of the process. So it was always very interesting to me, because I said, okay, if you have T cells that actually play a role in this feedback loop of, producing a lot of cytokines, and this leads the keratinocytes to hyperproliferate, and then it's kind of a self-sustaining loop, right? So now you take a treatment, which can be anti-TNF, and you see a decrease in T cells, and your skin looks like normal, healthy skin while under treatment. And I was like, but it's interesting because once you take the treatment away, it comes back at the same spot. So I was like, it seems like some T cells seem to survive. So I was interested in really understanding, why do some T cells survive, or what is it like? There is something that seems to remain, that seems to be different than the rest.
Can I just jump in there with
Yeah.
one thing? So there could be this memory. Could there also be some kind of selection process there? Does psoriasis that comes back on that same spot, does it get worse with, like, if it keeps repeating because you keep maybe selecting the strongest T cells or something like that?
Honestly, I have to tell you, my answer is, because I'm not a dermatologist and I actually don't see the patients, it's a very good question. I never thought about it. From what I know and how I would answer it at this moment would be, I don't think it gets worse when it comes back. So but it's actually interesting to kind of think about that you have an enrichment of certain T cells that then are even more active and cause a even like higher inflammation. So the answer is actually, I don't know. It could be. It's an interesting thought. So I was interested in understanding, is there a different type of subsets of T cells in the skin?
Okay.
So the way I approached it is by single-cell sequencing. At the time, I think I can call myself an early adopter almost. It was the very first version of single-cell sequencing back in 2018. Yeah,
Okay. Back when you couldn't just pay a company to do it for you, right? You had to...
Exactly. You couldn't just pay a company to do it for you. You really had to learn it yourself, and it was hard to learn because few people knew how to do it at the time. I was very lucky because there was a good, already a single-cell sequencing core facility at Karolinska Institute. So this woman that worked there, she really helped me. And there was a key moment when I've set up this experiment, and I think it's just important maybe to share for the audience. And I will never forget, when you do the single-cell experiment, you want to really kind of when you isolate T cells from the skin, it's very tricky actually, and you need enough cells to actually input into the single cell sequencing machine. So I optimized and optimized and optimized and optimized, and I would have probably optimized for another 2 years, but then my supervisor was, okay, you go now, you do this, you got this. So the funny thing is, I just experienced how things were never good enough for me, and how I would have just kept going and going and going before I do the actual experiment, because these single cell sequencing experiments are so expensive, and I felt like, oh my God, I just want to do this perfect. So there is no perfect, just do it, look at the results, and then adjust things.
It's
this
at
is
some point
one
you
of
have
the
to let
key
it go.
learnings.
Yeah,
you have to let it go. Yeah. So I used actually single cell sequencing at the time because it would allow me to actually phenotype a lot of different or discover potentially new phenotypes of T cells within the skin, versus if I stain with antibodies, right, and do a flow cytometry staining and run it in effects, I'm very not biased, but like I can only look at the markers that I actually include, which means I actually already have to have an idea what I'm looking for. So single-cell sequencing really opened up for me to look at different subsets or populations of T cells within the skin, where we're mostly interested in CD8 T cells. And Kina, indeed, we did find a little subset, which was really interesting, which did express CD62L. So if you're a T cell biologist, you know that CD62L is usually something that is expressed on lower differentiated T cells that have a higher potential for proliferation and replenish other subsets. So I was like, this is extremely interesting because it has been described in the literature before, but people always kind of disregarded it as like probably contamination. So if you think about the skin, you have the dermal layer and the epidermal layer, and within the dermal layer, you do have blood vessels. So there is actually, you can have blood contamination. So basically T cells that were in the blood vessels at the time of sampling that can end up in my sample. However, interestingly, the epidermis doesn't have any blood vessels. So it's completely cut off compartment from the rest. That's why I always love to work with the epidermis, because it would give me an insight on truly resident T cells in the skin. So, and I found in the epidermis, I found these CD62L positive T cells, and I knew they cannot come from the blood because there is actually no bloodstream going in. So I had a hypothesis. So could it be that these CD62L positive T cells are lower differentiated, have a higher proliferation potential, and can actually acquire the phenotype of something what we call a tissue resident memory T cell in the skin based on markers? I'm just throwing it in now. You have different markers, CD103 and CD49A. And the interesting thing is, so I set up all of these experiments and I cultured these CD62L positive T cells. I sorted them out from the skin. I cultured them on fibroblasts to give them like a natural environment. And then actually I looked at the proliferation, and they indeed did proliferate better. And I was like, yes, this was one of the key experiments, Keenan. I will never forget this day. I had this hypothesis and I was like, yes, it was the best feeling ever. Like, I cannot tell you. And I was like, okay, cool, interesting. So now let's see, when they proliferate, do they change their phenotype? And indeed they did. So Keenan, they started upregulating CD103 and CD49A, what a bona fide TRM cell in the TRM, sorry, I always say TRM, it's a tissue resident memory T cell, what a tissue resident memory T cell in the skin would express. And this is like where I was thinking, okay, cool, you found a subset that actually has the potential to replenish higher differentiated states. So this is basically the finding of my PhD, and these T cells can acquire all the different, so there are different phenotypes within the skin, and it can acquire all the range of phenotypes, has a higher proliferation potential, which means if you think about skin diseases, you might reconsider who you actually target in the therapy. So you might actually want to target T cells. Let me just call them a bit more stem-like, because people are more familiar with the stem-like term. So imagine these are T stem cells or memory stem cells. Memory stem cells for T cells. If you would actually manage to eradicate them, it's my new hypothesis, which I will never prove because I'm gone. You might actually be able to fully eradicate the disease. But that's, a new hypothesis that's out there now. But yeah.
That seems like a very directly applicable result. Was there ever interest? I know KI is Karolinska Institute is connected to hospitals, right? And then you get these samples. So that's a very good thing that you can more directly make this translational impact, let's say, with your research. Was there ever a discussion in your lab, if we discover this could be a patent, and then maybe we can, apply for something there and try to get some money, start a company, maybe? Was there ever, is there an entrepreneurial spirit, let's say, among academics, maybe in your field or at KI in general?
So I think there is an entrepreneurial spirit, because I know about a lot of groups that are at KI that run their professors, and they also run companies based on their discoveries. More specifically within my group, no, because I think it's about, the interest. So I think my supervisor is just very interested in science. That's all she wants to do. Science and science. She just wants to make an impact. She's not very interested in anything that has to do with commercialization. And to this point, I mean, it would have been hard to patent anything, because right now we're talking about a cell type. So what we would need to patent or potentially go forward with any type of commercialization would be probably to find something to actually target these T cells. And I think, it sounds like it could be very close, but I think it's actually quite far down the line, because CD62L in itself is a molecule that is expressed on a lot of different T cells, so it's not very specific. So I think the next step would be really to find something to specifically target them. And I mean, we are actually, I'm gone, but the paper is not published yet. So we're looking actually at a different marker, a transcription factor that we found is upregulated in these T cells. So that's still happening while I'm gone. And they're actually trying to stain now via flow cytometry, because we do see a transcription factor that is up in the single cell sequencing data. But, the mRNA doesn't really necessarily translate into a protein. So, this is one thing that we're still kind of following right now. Is there a specific target, or is there something that is specific to these T cells to distinguish them, or, kind of start finding potential targets.
I wonder how many more therapies would be available today if more scientists were interested in more things than just science, such as making a company. I mean, I know that some university, I mean, I went to a master's called Translational Medical Research, where we got actually a couple of, solid hours of statistics of how many, how many discoveries are actually laying around without people just didn't, the translational impact did not catch up. And that was the whole point of the master's. Here's a lot of basic research, and we need to educate a new generation of scientists who will actually, when they find something, be excited about, maybe making a company and have already the knowledge about it. I know Stanford, for example, has made a big hub for entrepreneurs who, they make a discovery and then they really support you. If you think you have something promising, if you could pitch it to the board and say, I have a really good idea, we have some solid results, they help you try to translate that. So that's pretty cool. I like that discovery. That's very, very interesting. Okay, so that is your baby. How does it feel to let go of your baby? Now you've recently graduated, and I guess the baby's not yet thrown away because the paper is not yet published, right? But you recently graduated. Congratulations. Tell me, how does it feel to graduate and slowly leave that world?
So beginning of 20, I had a hard time to let go of my baby, to be honest. It was very hard for me, because you work on something for 5 years and you will not bring it to the finishing line. At the end, I mean, it's good. I have a colleague. She will be the shared first offer with me, so she's now taking care and managing all the things while I'm gone, which I know I'm in an incredible. I'm incredibly grateful for it, because a lot of people don't have that, luck that they have someone who will actually finish it. However, I also think my supervisor has a huge interest in getting this paper out, because I think for the field it might actually be quite interesting, or like they might actually change the way they think about certain things. So I think it's super important to get this paper out. So she has an interest, so I know it will be out, which makes me a bit lighter. But I mean, in the beginning of 23, when I decided, okay, I'm going to defend at the end of the year, I didn't feel ready. I didn't feel ready to leave. I didn't feel ready to leave my project behind. I just felt like I didn't feel ready to leave this life behind, because, I've worked in this same lab for, at this point, 6 years. And at this point, it's just like, it's your life. You have your friends there. You have your work there. You have so many things that you share with, not only like in terms of your baby of scientific discovery, but also like private life, friends. It was very, very scary for me, to be honest. But as I kind of got used to the thought that I would graduate now, things slowly, I started to accept that I will actually graduate, and it became better, and I felt like, okay, I'm ready. So it took me, I think, almost half a year to feel like I'm ready now to leave and move on and start my own life, although I knew something so exciting was waiting for me on the other side. It is so scary to leave this known and jump into something where you have no idea what's going to come on the other side. But yeah, so it was very, very tough. I didn't want to leave where I was for multiple reasons, because of my colleagues. I loved my project. I really loved working on it. And yeah, then at the end, I mean, it's also a gradual process, You kind of, from the day when you decide you're going to defend till you actually defend, you got some time, and you just get used to the thought that things will be different. And now I'm starting to kind of accustomed to my new life, which is also pretty exciting, but different.
Okay, so your next move was to start your own company, and we will go there, and definitely want to discuss that more. Just before I go there, so I don't forget. So even though your main project's still not out, you've been an author on a couple of papers from your lab, and I just want to discuss a little bit the process that your lab went through in publishing papers. How do you choose a journal? How do you figure out when the paper is done? Things like that. So maybe tell me about, take one paper as an example and maybe guide me through the whole process from, okay, maybe it's time to start a manuscript now to start writing. and to submit somewhere? And how does that whole process go?
Ooh, that's actually a good question. So I have to say before I jump into this, I have not published my own paper. I am a shared first author, but there, like the first author, Beatrice, on that paper, she took the whole lead on, finding the journal. Of course, we discussed all of these things together, but she was the one actually doing, preparing the manuscript for a journal and so on. So I was involved in that process, but I didn't do it yet. So this is what I will face with my own paper, just to clarify this. But I think the thing, let's talk about my main project that is not published yet, because this is really the paper that I know the best. I have been a co-author on many different papers, but you're never as involved as when you drive the project. So when I started off the paper, we started with single cell sequencing. It was quite a safe play of a project because single cell sequencing at the time, we knew something will come out that we can publish, right? I always, I should mention, I'm this type of person. I would have rather had one big paper than 4 small papers. And not because of the impact factor, but because of the impact you can make with the work. Because
The real impact factor.
The real impact, exactly. If you commit 5 years to one project, of course you have side projects, but you follow one question and you dive really, really deep. This is what I wanted to do. I didn't want to split this into different papers. I just wanted to have something where it's a complete story, right? So it's like something that actually provides value beyond I just replicated something. I wanted to create novel knowledge. This was my thing. You never know how it works out. I mean, but this is the mindset that I went in with. So we started with single cell sequencing. We didn't really know what it will give us, but it was, as I said, a bit safe in the sense that it was relatively new and you always get something out of single cell sequencing. It's interesting. I mean, there is always something you can discover or find. I then further the process, the way it went for me, I discovered this cell and I knew, okay, these are the steps that I need to prove in order to make this publishable and make a point. So I knew, okay, I need to show that they proliferate better. I need to show how they distinguish themselves from all the other cells. I needed to know, can they actually acquire the phenotype? So there were certain questions that I needed to answer to make the scientific field see the impact of my work. So, and that you have a certain time frame. In my case, it was like, okay, many people do it in 4 years, and I did it in 5 years, the Ph.D. So I kind of set this 4 years time frame in my mind. So this is what I need to do in that time frame. And I mean, of course, you never know if you can establish it during that time, but I had a good feeling that I could do it. So maybe it's good time management or good project management in terms of planning and thinking. I guess you need to be open in science that results will go a different way and hypotheses are not true. And then, you come at a point where you need to decide, okay, we couldn't prove this. So maybe we will not actually be able to show certain things which will lower the impact of the work. In terms of now impact factor, where do we want to actually publish? What do we want to go for?
Because now it's a little bit of a more boring story if you did not manage to create what you initially wanted.
If certain things are missing to really prove a point, it's just not as impactful in terms of, let's say, I could never show that these cells actually proliferate better than the others. This is just such a sign of lower differentiation and the potential to renew. If I could have never done that, I would have had a hard time to sell this as a progenitor cell, because this is just a very important thing and I couldn't know at the time. So even though I always had this in my mind and these are the experiments I need to do in order to come to that point, you never know, right? For me, everything kind of went well-ish, let's say. And I actually managed to do the key experiments that I thought were important. To answer your question, you can always keep going, right? You can always dig deeper because now we have proven the cell type exists. They're different than all the other cells. They have a different functional behavior. They also produce different cytokines, which you would associate with lower differentiated T cells. This was also something I wanted to show. So I wanted to show they're functionally different when they're in this state where they express CD6012 versus they're then different when they start upregulating other markers that are associated with these higher differentiated bona fide TRM cells. They start kind of reassembling the cytokine profile of a bona fide TRM cell. So it's just, there were a lot of things that I knew and that I needed to prove. I could prove a lot of them in order to be able to go higher impact. So now we're at the stage where we're discussing impact and where can we really go with it. We do know that certain things are maybe missing. So for example, we have a lot of indications on a transcriptional level how these cells could be regulated through transcription factors. However, what we're missing is, for example, to make it more impactful, now you could say, let's do a CRISPR knockout. And let's see, how does this gene affect the cell? So we could now just keep going. But in the interest of time, and also knowing it's a good story as it stands and it will contribute to the scientific field, I think we made a point with the experiments we did. So we decided this is it. After 5 years of work, we will submit because for us, we evaluated it's good enough. If we wanted to go higher, we could keep going, of course. So I think it also breaks down to your, sometimes it's just more, does the lab need a paper? So you just have to push it out. It's not always like what you really want. So I had this time frame, and from this time frame on, after we had done the experiments, we decided, okay, this is it. Currently, they're doing some last experiments, but we're writing it up. We have sent it out to co-authors. So we started writing the first draft of a manuscript. I started writing, Kenan, to take you into that process in, let me think. It was the first draft I've sent to my boss written was beginning of 2022. That was the very first draft of this paper. Now I think we're at draft 13. We change the figures all the time. So the way it basically goes is we have the data. Now it's all about tweaking the story, right? Because as you know, you look at the paper, you read it. Most likely the experiments were not done in the order as they are presented. So now it's all about what is the most impactful story that we can write around these, around the data we actually got. And that's just really about understanding what's the gap in the literature and how can you actually kind of embed your own research within it, and to make actually also, other people see your impact, because the reviewers, you want to kind of convince them you have done something great. So it's all about phrasing a story that will convince them that this is great and that this is helpful for anyone else in the future. And yeah, we're still working on it. We're still kind of like moving figures around, should we do now? Should we switch figure 4 and 5? This is kind of like, what is a better flow? How is it more convincing? We have all these like discussions on this level right now. Yeah, so it's funny because once you're done generating the data, you move into a different phase where you just go back and forth with a lot of the co-authors and you discuss, what do you think? How could it be the most impactful? And yeah, and then honestly, for the decision of the impact factor, my supervisor, like which journal to aim for, she always reaches out to, like collaborators or colleagues that usually also review on a very high level in other journals. And then she just also asks for feedback. What do you think? Where can we aim for? So this is a little bit how we decide, because I personally am this person. I would always say aim a bit higher than you think you can go, because you never know. I mean, it's just like, you never know who will review it. You might be lucky, you might be unlucky. I do think you need an amount of novelty if you want to go high, of course. And you need the experiments that actually prove it from many different angles. I think that's also the thing with the higher impact, that you have, not just one experiment hinting in a certain direction. You have multiple experiments that hint in the same direction that that your hypothesis is actually true. So yeah, so we're trying to aim high-ish, I would say, with this paper. Whatever that means, because it's different for different fields, right? If you talk immunology, what is high versus if you talk pain, it's a very different impact factor. So yeah, in pain, I realized impact factor 10 is really high. If you go in immunology, you're probably talking 30 or 40.
Mhm.
like immunity or nature immunology.
Just because the field
Or
is so big and
yeah, exactly. The field is so big. So yeah, so I mean, this is where we are right now. And I mean, we haven't really decided on a journal. I think it depends really on this last experiment that is being done right now, because showing there are sometimes these small experiments that actually, as I said, the proliferation, it was super necessary to make a really strong point. And I think the experiment that is being done right now, it can increase the impact factor, because all of a sudden we're not talking about a generic marker that is expressed. All of a sudden we're talking about a more defined marker for the subset. And that's, of course, a bit of a different story. So, yeah. I don't know if that was in any way clear. I feel like it's a long process, and
I think it's a great answer, and especially I think the point you made about about deciding the journal that you submit to. Your professor often reaches out to their colleagues and discusses, because no matter how big the field, it's still kind of a niched group of people within your field that can say, this is actually a very big discovery. And there's a limited number of people who can say how big the discovery is, because I don't know what's the gap in your field. And most immunologists probably don't know what's the gap in your field besides the couple of leaders, right? So that network is, in the end, there's a couple of people that stand at the top of the mountain that is some niche of a field, and they kind of decide how valuable something is. You went over one thing that it sounded kind of trivial, but to me is not trivial. And you mentioned in the beginning, when you were writing your process, you were writing your paper or doing your paper, you had some things in mind you wanted to prove.
yeah,
And then you had a time frame, and you just kind of glanced over, you have to be open to your hypothesis might go right. But I don't think that's so easy. I think that's a very difficult point to reach as a mature adult scientist, to have your baby hypothesis that you invest in so much. You try to do several experiments to see if it works, and it would be very exciting if it works, and then you just see it just doesn't work. So there's naturally, I noticed in myself, a bias that I have to fight. When I see a result that is negative to what I want to see, I have to really fight. There's a piece of my heart that leaves, and I have to say, no, this result is shit, and I cannot believe this if I want to build on top of it. So how did you manage to fight against your own human proclivity to this kind of bias?
it's a very good question, Keenan. And I think it was my supervisor who really just taught me we're doing science. We're not, I mean, it's not we're producing something and it's always true. We have to always be open for whatever we do. It might not show what we want. And she always normalized this feeling. She always said it's completely okay. This is what the data shows. And then we move on. It's hard to let go, though. I mean, I tried to figure something out. I mean, Keenan, I think I tried it for 1.5 years until we said, okay, we're switching course now a little bit. And it changed the story of the paper. It changed a lot of things, but the reality is it didn't work. So I couldn't do anything. And I think it was really the environment that normalized it for me. I was just like, I felt super supported in that sense of, because I know how it is. You really want this to be true, because you think if this is true, this is just going to be so amazing. And then you want it to be true. And then you're actually also biased. And I think, thinking of whatever you do, having a hypothesis, but just being completely open, because, even if it's not true, it's still a result. And that's also interesting.
Mm-hmm.
And, the important thing for me, Keenan, in this sense is always like, I'm just saying to myself, I wouldn't say it's generally not true. I would just say the approach I used to test it did show this. I mean, it depends a little bit on the context of the experiment, because, so for example, for me, it was like I couldn't make an experiment work really. And it didn't show me the result I wanted, but that doesn't mean if you took a different method, maybe you could have made it work and you could actually have proven my point. It's just about, and maybe with the technology that is currently available doesn't allow you. And maybe, in 10 years, someone will go and do the same thing and be like, I have all this technology now and I can maybe prove it in a different way. So it's just, yeah, I don't know. It can be hard, especially if it's very fundamental for your project in order to move in a certain direction. Yeah, but I think that's something, it's a hard part about science. And sometimes, it's research. So we're trying to search for answers, and they're often different than we expect them to be, which is exciting too, in a way, I think.
Very, very important that you mentioned how your supervisor normalized this feeling that this is what the data shows, and we should not get attached to it emotionally too much. I have a quote. I have two quotes actually that I would like to share now. One is it's kind of our version from my lab, like how my professor normalizes that environment. When he sees data that looks too beautiful, he says, this is too beautiful to be, probably too beautiful to be true. So we will, so we always joke about, last lab meeting, someone had a very interesting discovery, but experiment needs to be repeated. And my supervisor goes, that's very beautiful, but as we know from the history of the lab, only about 20% of those can be reproduced. So let's keep going. And then another quote was very interesting from a postdoc from our lab. He said, yeah, that's why science is interesting. We tell each other stories until one of them turns out to be true, but most of them we will have to trash. But yeah, so it's just a bunch of stories that you have to kind of selecting the true stories. All right. So I love that answer. Thank you for that. Okay. So I do want to switch now to your current life. What are you doing now, and why are you doing it? Not at this very moment.
No.
Career-wise.
What am I doing? I mean, so well, since the beginning of the year, we have registered, like me and my co-founder Angela, she's located in the US. We have registered our own company, which is called Alma-Me, which aims to help PhDs in their transition from academia into industry. And how did I actually come to do that is while I was doing a PhD, I was, involved in different organizations. I've always felt a little bit isolated when I was sitting in the lab. I don't know, you might have already realized by now, I'm more of a people person. I'm social. I love to talk to people. I love to connect with people. So, I realized during my PhD, everybody was always like, I don't like lab meetings. And I was like, I don't understand why everybody hates lab meetings. So for me, this was like a time where I flourished when we came together and we had coffee and, we discussed science. And it was just like I was part of a bigger team in that moment. Whereas when I sat in the lab, I was often alone. I had sometimes very early mornings, late evenings, like we all know, and I felt isolated. And I realized it's not for me. So I became part of different organizations just to do some extracurricular stuff to broaden my horizon, of what is out there, but also to start building a network, to get in contact with other people. And one of the organizations that I joined was called PhD Careers Beyond Academia. And I joined it a little bit for a selfish reason, because I also really wanted to know what can I actually do with my PhD after? Because we really seem to tend to focus ourselves so much on the subject matter expertise that we acquire. But when I ran these seminars, I had people that went from PhD to McKinsey and became a management consultant. I had people that became marketing managers, editors, research equity analysts. And what I realized, what they all had in common when they told their story, it's like you do have a subject matter expertise, but you acquire a ton of other skills during your PhD. And it was just like, for me, it was like, hey, they're right. You're right. You you present, you communicate, you solve problems all the time. This got so stuck with me when Zach from McKinsey was there, and he said, PhDs are great management consultants because we find creative solutions to problems. And I was like, you are right. It's like I just started realizing there is this wealth of skills we bring to the table. And I realized if I didn't know, other people also don't know. So I gave these sessions, and during these sessions we had PhDs attending, and I got also to talk to them and chat with them. And I just loved it because I always left these meetings very, very fulfilled. There was something inside of me that just showed me in a direction. This is something maybe you should be pursuing in some sort of way. And then I was like, okay, I also have this entrepreneurial spirit inside of me because, Kina, the way I work, I love, and that's also why I love to do a PhD. I love to do the PhD in my group because I knew my supervisor would give me freedom. I had freedom to operate. She had a good amount of money, which gives you a lot of freedom in science. I mean, it is a fact. If you have money, you can do exciting experiments. Well, you can also do exciting experiments if you don't have so much money, but it's just like you're not limited by, oh, can I do this experiment or not? Or do I need to think twice? I never was in that situation.
Exciting experiments are way less likely to work. So that's why it helps. All right.
Yeah, exactly. But you see, and also I love to think outside the box and come up with creative solutions. So it's like, I have this part inside of me that loves freedom, that loves to come up with creative solutions. But also I love to help people. I love, I'm a PhD myself. I know what it means to go through all of this. And I was like, I kind of think I could build a company where I could live all of my strengths bringing into this company. And then I had a very fundamental moment, which I would everyone recommend, and I will just share this now because no matter if you want to go to industry or if you want to build a business, I had this person, Samer. He was giving an entrepreneurship course and he said to me, Elena, you will not go anywhere without a network. This is what he said to me. And I was like, okay. And then he made an introduction and then I joined another organization. And through this, it all started like kind of developing in the right direction. But I always had a focus on this is where I want to go. I knew where I want to go. So I knew, okay, I need to get into certain networks to get where I want to go. So I started getting into founders networks, actually through LinkedIn. And that was, I think, end of 2022 already, where I started networking myself in there, just so you get a little bit of an impression of the time that actually has passed until today. I started end of 2022. Not actually, it's not true. It's probably, on LinkedIn, I started end of 2022, but the actual process of me thinking, establishing a company, building a network more locally started in 2021, end of 2021. So 2 years, approximately 2 years ago. And then I just put myself out on LinkedIn, network myself into founders networks, got to meet crazy people. I cannot even tell you. Crazy people.
So this
I then
mostly scientists who went into finding companies or
no,
people from all walks of the...
People from all walks, because also my project wasn't very scientific. I did a lot of other things. I also went to Karolinska Innovation Department. I did so many different things actually I really wanted to pursue this, and this is the thing, when you really want to do something, you don't see challenges, you just find solutions. This is the interesting thing. And this is, I think, what makes some people more successful than others, because if you know where you want to go and you are passionate about your mission, you will always find solutions. So I went to so many different places just to find a way of moving forward, because I can tell you, when you think about building a business coming from academia, you have no idea where to start. It's just so difficult because you come from an environment where no one shares your passion or is like really, thinks in the same way you do. It's just a different mindset. And so I networked myself into this founder network on LinkedIn. And through this network, I got to meet my co-founder, my today's co-founder, Angela, which is an incredible woman with like 20 years of business experience. She's actually working as a consultant on the side and consults other businesses that don't kind of like work, to help them, fix their businesses, to get them back to revenue or whatever their problem is. So I think she understands business as very few people do. And I feel still very honored that we get to work together. So, and the way I see her and me a little bit, I'm like, to just be a bit more, to give an analogy, I feel like I'm a bit, the Steve Jobs of the company. I'm a bit more in the front. And then you have the Wozniak, which is Angela, because, I'm this person. I love to network. I love to build relationships. I love to go out and speak and inspire people. And Angela, she loves to fill in Excel sheets, which is like a horror for me, So she really manages everything in the back end. Exactly.
From an Excel sheet. Okay.
Exactly. So, and actually, Kiran, I think I'm not even sure I answered your question. It's so funny because you asked me where I am today and how I got there. But the long question is, this is how I got here. And yeah, running this company now. I'm calling myself a CEO, which I think is strange to myself. But yeah, this is where I am
That's pretty
at the
cool.
moment.
So what
Yeah.
does it mean? I think we as scientists don't get a lot of exposure to words like networking. And I
Yeah.
feel that there's a little bit of an undertone of that's for business people, you know. That's not for us scientists. We
Yeah.
just do our work in the lab. And so when you say you networked yourself into something, how does that look like? Like, what does it mean to network with a group of people? How do you find these people that share an interest that you would like to peek into?
So I think, first of all, the way I would probably describe networking is just relationship building. You know, Kenan, it's like us talking. You know, we know each other, we support each other on LinkedIn. You know, you comment on my post, I comment on your post, you build a relationship. You're much more likely, if I need a favor from you, you're much more likely to help me, right? It's just the way it is because you know me on some sort of level. And that's all networking really is. It's about building relationships. And how do you build relationships? It's trust. You need to trust. How do you build trust? By being consistent, showing up consistently, maybe, you know, commenting consistently, showing support consistently. And this is all what networking really is. And I know it's hard, but I think going in with a mindset of just being curious about other people and wanting to understand what they do can open so many doors without actually expecting anything. So this is the biggest misconception I think we have in academia, is that we go out and networking is asking people for favors. No, it's not. You can ask people for favors at some point down the line when you have established this relationship. That's why I'm saying I started building 2 years ago, because this is what it takes to build relationships. It's what it takes to build trust. You don't build trust overnight. You build trust by consistently showing up, consistently showing your support, consistently meeting in some sort of way, showing interest, genuine interest in each other. It can look like you go to certain events, like you, for example, here at Karolinska Institute, they have at the KI Science Park, they have events where different companies come together. Go there, not only once, go there multiple times. Just have normal conversations. Don't ask people to do anything for you, because that's not what networking is. It's just about, you know, talking about private things sometimes, talking about struggles, like relating to each other. And it's the same, then it translates if you go to LinkedIn. I mean, just start commenting on people that are relevant to you. So, and this, the whole point with knowing who is relevant to you means you need to know where you want to go, right? So if you don't know if you want to work in pharma or in consulting or I don't know where, it's very hard for you to do a more targeted networking approach. So I knew I wanted to become a founder, so I focused on founder networks. And the way I started, Kenan, I didn't write a post until June 23. All I did was commenting. All I did was engaging with other people on LinkedIn. And when I did write my first post, a lot of people supported me because they knew me. They knew who I am. I engaged with their posts. So it's all about, you know, and then at some point people say, hey, why don't we have a chat? And then, you know, you meet like we now today, and this is like what really builds trust. You get to meet the other person. You like the people, because at the end of the day, I mean, if a stranger asks you on the street, can you help me get a job? I mean, no, clearly you cannot. But if you have this trust and this relationship, then you can say, hey, I see you work in this company. I'm interested. Could we maybe have a chat? Or would you mind maybe, you know, having a look at my application? But you need the foundation to ask people certain things, because otherwise they will not be willing to do it for you.
How would you recommend for someone who's in on more of the introverted side of that scale to network? Is there something that you can do as an introvert to get yourself going in that space?
I know. So first of all, I think networking, introvert, extrovert, it's always out of the comfort zone. I think I have a very easy time approaching people, but I'm still, you know, for me, it's very far out of my comfort zone. I think the important thing is when you go to networking events, for example, remember everybody's here also for the same reason, right? They want to get to know people. They're not here because, I don't know what. They're here because they also are open to meeting new people. And I think if you're already aware, okay, we're in the space where people want to meet new people, it's easier to maybe go and approach someone. But a really good opener always is you go to an event, you have a speaker, and the speaker talks about something. First of all, asking a question can always help because this person will remember you. When you're done, let's say the speaker is finished with his talk, and then maybe there's a mingle after, you can always approach the speaker and just compliment them. It's such an icebreaker and say, hey, this really resonated with me. Or you always have an easier time if you can find a common ground, for example. Which is easy when you're in science and you go to conferences, you just talk about your science. I mean, that's a common ground in a way. Or, oh, I see you have been at Harvard. Me too. You already have this commonality. And this is like when you go out and you want to network yourself into business, find this commonality. But if you don't find a commonality, make a compliment. Just say, hey, I really loved your talk. And I actually had a question about this, or I wondered this, or whatever. And just try to break the ice. And then remember, everybody else who is at the event has some sort of interest in the topic or wants to meet new people. So I think it's about normalizing the thought of, I'm actually approaching someone and maybe they don't want to talk to me. No, just be open and assume people are open too. And just approach them as a human being. I think that's the really only important part. And if you manage to break the ice by, again, Keenan, how it is. I mean, if I tell you, hey, I have been at Harvard too, immediately we're like this, we have both done a PhD, and this is something PhDs need to take advantage. Having gone through the PhD experience makes people want to help you. If you reach out to someone who has a PhD, just because you're doing it yourself, it will make this person want to help more, because it's a common experience. When you look at PhD memes, we all understand them. We can all relate to each other. It's about relatability. It's about, all of these things. Humanness, honestly, just be a human when you network.
Oh, it's so hard. No, it's, well, I think it was, okay, so finding common ground, very, intuitive, but, also practical. And another thing to understand is that when you go to an event like this, which is something that I experienced recently going to a conference, that was a Drosophila conference. So a bunch of, I think, 800 people from all over Europe, just working with fly scientists. And afterwards, there was a poster presentation. It was 300 posters, many, many posters. And I think I just went from one poster to the other and just asked, tell me what you do, And I've never met so many people who randomly just wanted to excitedly talk to me all at once. So understanding that when you go to an event where it's set up for that way, so actually can be a breeding ground for you guys to talk, right? So people will be ready to talk to you. That's why they came there. I think that's very important to understand. Okay, cool. So one thing I want to ask is, I think, I don't know if other people think this, but I have this conception that is very likely a misconception, that as a scientist, even if you go to industry after you transition from academia to industry, you can still just be a scientist. So you're just being a scientist and getting more money, and maybe you're more constrained with whatever the company's doing. But there's so much more than that, right?
You mean in terms of
just so much more you can do when you say go to industry. That doesn't mean now you pipette for a company. You can go to such a big landscape, right? Tell me more about that.
I think so. This comes back to really that a lot of PhDs focus on their area of expertise, right? I know how to pipette. I have some sort of background in immunology. This is where I should be working in, which kind of classifies you maybe for pharma, in terms of industries, or maybe biotech. And maybe you think of a scientist role where you can pipette. But what really opens a lot of other positions to you is understanding all the other skills you have acquired. Because I always say, The world is your oyster after PhD. In my opinion, you can do anything. And I think it's exciting. And this is also where I want to get people. I want them to feel I have gotten this degree. And instead of feeling I have nothing to offer for the world outside there, I want them to understand you can literally do anything, right?
Wow.
I mean, of course, there are certain roles where you need a technical expertise. If you don't know how to code, it will be hard to become a data scientist. That's unfortunately the truth. So if you want to become a data scientist, I guess you have to upskill in this case. But, let's take the example of becoming a management consultant. You do have your subject matter expertise, but what is really important in being a consultant? So first of all, they will teach you a lot of things when you go in, but what they really need is people that can think outside the box and solve problems, right? Creative, problem-solving solutions that are able to communicate. Well, maybe, they know how to present, which we have done. And, you do work with some Excel sheets, as I have understood, when you, for example, move into management consultant. So you see, if you only focused on immunology and like maybe, your flies, you would narrow down your employability a lot. Versus if you also kind of start understanding, you can be a scientist, but you could also be a project manager, managing other science projects where you can still be connected to the scientific side, but you're not actually doing the pipetting anymore. So, there are different nuances. So there is a scientist where you actually pipette, but then you can be working in project management where you manage scientists, you still are connected to the science, but you're not actually doing it anymore. So there are multiple layers of things that you can do, but you need to focus on other skills than your area of expertise. I mean, I had someone once at the at our talks. I mean, she's a marketing manager. Of course, she was in a biotech company, so she had some understanding of the products from her subject matter expertise. But all she did was marketing, because you need to understand in marketing. You need to understand who's your audience. Who are you addressing? If you're a PhD, you know your audience. It's the same for me. I'm a PhD. I know my audience. I know what I need to write, what resonates with them. The same with you, Kenan. You have gone through it's like, it's just so, there's so many other things. If you love writing, go into medical writing. You have been writing so many texts, and it's just about, I encourage people to think like, when you do a PhD, for me, it's always like, I compare it like running a small business. You go through so many different things. You do administration, right? You do experimental planning, you do execution, you look that you have the resources, so you order your stuff, you analyze it, you present it, you communicate it to different audiences, you kind of sell and market it, right? Like there is nothing you don't do. Sometimes you even need to pull in money. You need to write grant applications, you write papers, so it's just like, you do all of these things. And if you pay a little bit of attention, I'm always in a flow when I write, then maybe I want to go into the writing thing. So this is what I want I want people to be encouraged to feel, when am I in my flow? Because you go through all of these different tasks during your PhD. And remember, the world is evolving very, very fast. I think one of the most important skills you can bring is the ability to learn. And we all have it. We all started at a point where we were clueless, most of us at least, felt overwhelmed, and we managed to finish it with a PhD and now kind of considered to be an expert in something. And this is important to remember. You have gone through the process. You can do it anytime again. And of course, maybe if you're lacking a lot of skills, you will start in a lower position, because they actually have to teach you much more than someone who is already coming in with more knowledge. But just saying, I mean, I talked to a guy. He also did a PhD in data science, and now he works in a construction company. Because guess what? Data is data. Whether you're in a construction company, in a healthcare company, it's just like, getting this broader picture of, when you work with data, you can literally work with any data in any industry. So you're not restricted to a certain industry, and you're not restricted to certain roles. But it's something a lot of PhDs are not aware, and this is one of the things that I want to bring into the world, to make PhDs aware. You can literally do anything, and yeah.
That's a good vision statement, making PhDs believe in themselves. If I was starting my PhD, which just happens to be conveniently that I am, and many people
Yeah.
are in my position. And maybe I don't know where I want to go after. Okay, if I have a chance to become a principal investigator after, if I discover something amazing, maybe I'll stay in academia.
Mhm.
But most people, I guess, and more with as the time passes, because there's less and less academic positions, I feel, and the number of PhDs is just going up. When would it be a good time to start thinking about these things? I feel like it takes a long time to network, to find what makes you flowy, and where would you like to go into this landscape of industry jobs. When would it be a good time to start planning?
In my personal opinion, I think midway PhD. So, I mean, optimally, you would go into your PhD and maybe already just pay attention.
When the despair is the highest.
Yeah, exactly. Exactly. Optimally, you would just go into your PhD, and I know that's a wishful thinking from my side, but you would just be very kind of like aware of, these are tasks I like, these are tasks I don't like. I actually wrote a post about it. What I think can be very, very effective is sit down once a week and just reflect. What did I like? What did I don't like? And what do I miss? Because, when you think of, oh, I really love being in meetings like me, you need to ask yourself, why? Why do I love being in meetings? And it's because I can connect with people. Why do I not like to be in the lab? Because I feel isolated. And, I love creative environments. The lab is not a really creative environment. I'm a very visual person. I love good looking things. And so when you kind of understand, because the way I see it is, you don't go out and say, I have these skills. Which job can I do with these skills? You go out and say, I'm Kenan and I'm interested in X, Y, Z, and I have these skills. And I'm looking for a position that matches my personal interests, my values, and my skills, right? So as an example, I have run single cell sequencing for 5 years. I have done a lot of experiments. I would actually call myself an expert, and I'm sure there are a lot of positions out where I could probably just go and work in single cell sequencing because I'm an expert now. I know it in and out. But do I want to do it? No. And this is what is really important, because what I see a lot of PhDs doing, they go like, okay, I know how to image, I know how to this, I know. But what do you want, So understand what you want. And this, you can start at any time, the earlier the better, but I would really recommend, in my personal opinion, start mid-PhD, because once you understand, okay, wow, I really like to do these things. Let's say you want to be a medical writer. You realize I love writing, and I love to be creative and all of these kind of things. You can start building a network in that direction. Or you can start nowadays in 2024, you can start build a personal brand around it. When you walk out of the PhD, you might have a network of people that wants to support you, that know you, and you come from a place where you don't ask anything. This is always important. When you start building a network, it's better to start early where you don't actually need it. So when you come to a point that you need it, the relationships that I mentioned earlier, they're already established. So it's a no-brainer for people to be like, hey, yeah, of course, Kenan, I can introduce you to this person. Why not? But it's, if you ask someone you have never met in your life, your chances are really low that they will reply. Or, unless you have a common ground, like reaching out, asking other PhDs for informational interviews. If you do it like a human, or if you reach out from human to human, I think it works like a charm, honestly. So, yeah, I would say mid PhD, but yeah.
And if I'm not mid PhD, if I'm kind of panicking now at year 4 or 5, and I don't want to do a postdoc because this whole thing of doing lab thing, I just don't like it. I want to go into a company, do something different, but I'm kind of short on time, and time is money, so I should find something fast. How would you advise me to make a plan that is most efficient, that gets me somewhere, let's say in 6 months or in some reasonable time,
So, well, let's say you will probably not be able to leverage a network in this case. So I read an interesting survey from LinkedIn. They say 85% of the positions are filled for networking, which is insane. But one has to be realistic. Building a network that will actually help you within 3 months, or in this short period of time, is tough. It's not impossible, but I would probably bet on something else. I would really try to understand what I do have to offer to industry and really write a rock star resume that communicates your value clearly to industry and what you actually have to offer. It will still be tough, I think, to get looked at just because of the volume that goes through. I think a general tip is always, if you want to increase your chances for being noticed, you can do a couple of things. For example, if you don't have a network that can refer you in, apply as early as possible. Because you need to think of a company, when they get 1,000 applications, they might actually start going through the applications as they come in. If they find suitable candidates in the first 100, you might actually be out if you're candidate 900. Another thing is, you can actually also just reach out if the hiring manager is posted and express your interest. It's something very few people do, but you will be on top of the mind. But generally, I think your best bet would really be get a resume that will get you a foot in the door. Because I do see it, if you know how to communicate your value, Kenan. I mean, honestly, like, this is a real life example. We have a girl, she joined our course, I think 3 weeks ago. And she has applied for a lot of positions. I think for the past half a year, she hasn't gotten a single interview. So she took the course, she tweaked her resume according to what we tell her in the course. She also got a resume review from Angela, who has worked as a hiring manager for 20 years. They tweaked very little things, Kenan. And now she got an interview at a very big pharma company in Germany. So it's really about understanding how you can communicate your value. And if you manage to show, because this is where most of the academics fail. It's not that we don't have the skills to do the job. It's we do not manage to align the skills that we have acquired with the job description. And you cannot expect that any recruiter would sit there and be like, oh, Kenan has done all of these amazing things. How exactly can he now provide value in this position? You need to be the one to tell them how you bring value and how you solve problems in this position. And that's I think, my best bet. And do informational interviews, because this can be a really good way to kind of, how should I say, like, it's more of a short-term networking. I have heard of people that did informational interviews. They said they reached out to someone and said, hey, I'm about to finish my PhD. Would you be open for a chat? Just like 15 minutes or 20 minutes. I would like to hear about your experience. And sometimes they actually say, because you need to remember, most of the companies, they have something that's called a referral bonus. This means if I refer you in, I get actually money. So if, let's say, you have an informational interview with someone and they say, oh, actually, we have an open position, maybe I could give you a referral. This is something you can also do, for example. I think informational interviews can be a really, really great tool. And of course, LinkedIn, always LinkedIn. I mean, you will not get the same depth of network, but it's never too late to start putting yourself out, commenting with some very thoughtful comments, for example, on content creators that work in your niche or in your field. Yeah, because this is also how you can get recognized. I mean, if you post yourself, I think it's the most effective way, but it's really not for everyone. So I would recommend you also to just become very clear quickly of what you want to do, and don't go too broad, because this is what I see a lot of PhDs doing, Kenan, they apply for everything because they have no clue what they want and do not align their wants and interests with positions. And what you will get is a very generic application. It's
kind of the same issue that most students are going through when they're doing academic applications, right?
Yeah.
The shotgun approach. Let me just send 200 CVs to 200 generic letters and hoping for an answer. Sometimes it works, but then I tell, but do you want to get somewhere by accident, not knowing where you're getting yourself into, right? Wouldn't it be much better that you really understand what you want and where you want to go, and get some clarity on that, and then tailor 10, 15 applications instead of 200 to those 15 places. And then if 3 of them work, you would be actually delighted to get there and work there, rather than just, oh, okay, I'm getting into this company. I have no idea what I'm getting myself into, what kind of environment. And it's kind of a rolling the dice, with your luck. Maybe I have a lot of people in my, let's say, LinkedIn network and my newsletter that are not from Europe. And they often ask me, I have a couple of friends who are, say, from India, and they're trying to get a position in Europe in a company. And they're mentioning, probably for them, it's going to be a hassle to get me a visa and whatever. How difficult is it for international people, like people who are maybe from even another continent? And how can they overcome that if they're trying to, let's say, get into industry in Europe?
Honestly, I'm not an expert on this, I have to tell you, because I simply don't know how the visa regulations are or how willing certain companies are to go through a visa process. I can tell you from a perspective of what is happening generally in the working world is that people try to strive for diversity. So you want diverse teams. Diverse team means you actually need to be able to get people from all over the world to get just these very different mindset and views onto things, right? So from this perspective, I think, and it might be very company dependent, and also there are sometimes regulations in place within the country, so not even a company, but within the country, that makes it very, very difficult for people to move. So I need to think of something. So for example, if it's not very easy to bring a partner along, like there are other like kind of levels that one needs to think about rather than only the company level. I think it's also certain things. Now I have heard, for example, I have talked to it's not Europe, but just as an example, in Canada, in the French speaking side in Quebec. Now, for example, if you move to Quebec, you need to speak French. It's a new rule if you want to work in a company there, which obviously limits you to move there, but it's regulated on a much higher level. It's not even on a company level. So the reality is, since I have not too much, it would be something Angela could probably answer honestly better, because I have never been a hiring manager and I'm not really familiar with all the struggles. I do think generally for a company, it's of course more work if they have to go through a visa process. However, if you find companies, and I love companies that embrace diversity. So if you find companies that actually value this, I think you might have maybe an easier time for them to help you get a visa. I just know, so for example, if we Europeans want to move to the US, and I'm not familiar how it is in Europe, in the US it's very hard, because what they need to show is basically, the way I understood, when they hire someone that is a non-US citizen, they need to justify why they hire you. So why can a person in the US not do the job? Why do you need to get someone from outside? So I'm not actually quite clear on how this is with other companies. I can tell you right now, in Sweden, if you go to the immigration office, like getting a visa can take up to 6 to 8 months. So, these are the small practicalities that sometimes can get in the way of actually hiring someone maybe from outside, because maybe a company knows the actual process right now is very, very long. So I know from friends that need to get their visa renewed, they're now sitting here waiting to get a renewal, but it takes like half a year, so they can actually not leave the country during that time because they would not be able to get back. So I think it's a very complex question that I, first of all, don't think I have really the necessary experience to answer. And I think it might be very company dependent. I would maybe look for companies, if I were someone wanting to come in, I would look into companies that that try to be diverse, because they obviously need people with different backgrounds.
So to combine those, network with people from companies that value the diversity. That will give you that'll give you the biggest impact chance.
I think it's also, I think it might be so individual from company to company and from country to country. I'm not sure there is a really easy answer. Yeah,
It's a case-to-case basis, probably. Okay. So what is next for you? What are some plans for the, let's say, next year? What's happening in Alma Me, and what are some next things?
A lot of things are actually happening. So we released our first product, which is a 5-step course, which aims to help PhDs in their transition from academia to industry. And it really just gives you all the tools you need to be able to make that transition. Like, it helps you to discover, go through discovery. What am I good at? Because most of the people don't do it. That's the reality. So what am I good at? What are my values and how do they align with positions? And then in the next step, we kind of, tell you how you can start building a network, how you can establish an online presence, and then how do you actually align and communicate your value in a resume so that industry actually understands what you have to offer. We prepare then people for an interview, and then like how do you negotiate the salary and the how can you be an active member in the onboarding process once you accepted a position. So it's all like an on-demand course, which is a video course, which we are now currently, because I think it really builds a basis for every PhD student to understand how can I approach the process. So I would like to get it into, as a next step, into universities. So this would be one of my big goals, that I can actually go to career centers and maybe say, hey, look, you guys do really valuable work, and the one-on-one advising is super needed and I will never replace it. But your workload can actually be reduced if I do the basis of the actual work, and then maybe they come in with very specific questions that they are left with. However, also our program has with me and Angela, you have a half an hour Q&A every week, so you can come in to us with more specific questions too, where we can actually help you if if you're stuck somewhere, if you don't know how to phrase something in your resume. And you get access to the course community. So I really want to get this into universities. And I have actually taken the first step yesterday into that direction, because right now what we started with is basically literally just selling it directly to PhDs. But I would wish that these PhDs don't actually have to pay for it, but that the university pays for them and they can then just go through a university account and access it. So this is
at the moment, where can people find this course?
At the moment, they can find it actually on alma.me. www.alma.me. That's our web address. So if someone is interested, so our main goal is really to keep it affordable for everyone. So I think that was very important for us. So I think it is affordable for every PhD student. So if someone is interested, also like just happy to have a chat, reach out to me if someone wants to know more or hear more about it. This is one of the things, and a next step that we're actually looking into is because we realize these PhDs are really open to work because they don't know where to go, what to do. So now we are trying to build this bridge between academia and industry by giving them the tools to be successful in business and prepare for business, but then we also actually connect them with real open positions.
Awesome.
All
right.
Yeah, this is the next step.
So, Elena, you are very active on LinkedIn, so I know people can find you there. Is there any other place where people should go and follow you in your work?
Actually, yeah. I mean, I'm on Instagram. You can follow me personally under Ellie_the_scientist. That's more of my personal account. But then we also have an account which is called Alma Me Ph.d. It's something I'm just building up. That's a completely new thing for me. I mastered LinkedIn, I think. Now I need to master Instagram. So Instagram is a good place. We have a YouTube channel, actually, which is called, I think Angela called it Dr. Elena Hoffer and Angela Priest. PhD, from PhD to industry. So we do have that channel too. And Angela is setting up a podcast. So we're soon going to be at a lot of different places, but I think the local ways or the central place to find all of these other places where we're to be found would be our website, www.alma.me.
Awesome. Well, Elena, thank you so much for talking to me today. for sharing your experience and your journey from KI to making your own company. I wish you all the best. I hope people who are listening to this found it useful and interesting, and they go to alma.me and they check out your course. And I wish you all the best in your business.
Thank you so much, Kenan. I really appreciate the invite, and it was really fun talking to you.
Awesome.
And
all right.
yeah, thank you so much.
Till next time. All right.
Till next time.
And we